viewers, today we will be discussing on the topic translation. Translation is the process wherein the RNA gets converted into protein. Translation is supposed to be the most energy demanding process as because herein for the first time the information which is coded in the form of nucleotides gets converted into polymer of amino acids. It comprises of at least 80% of the cell's energy is utilized in carrying on the translation process, and we have seen that almost 50% of the cell's dry weight is involved in carrying on the process of translation. You will be astonished to know that to translate or to synthesize a single protein, there is involvement of 100 other different proteins. So, the process of translation mainly comprises of 4 important components. Number 1, messenger RNA. Number 2, transfer RNA. Number 3, amino acid tRNA synthetase. And the 4th one is the ribosomes, which is supposed to be the mega Dalton machinery wherein the actual process of translation takes place. If we compare the ribosomes of bacteria and mammalian cells, we can see that bacterial ribosome is of 70S, which can further be divided into 23S and 5S and 16S. It comprises of 31 and 21 proteins respectively. On the other hand, mammalian ribosome is denoted as 80S, which comprises of 60S ribosomal subunit and 40S. The 60S can be further divided into 28S, 5.8S and 5S. It contains 49 proteins, and the 40S ribosomal subunit is containing 18S ribosomal subunit and 33 different proteins. So, ribosome is basically a complex between ribosomal RNA and proteins. So, let us have a look that ribosome comprises of 3 different sites, namely the P site, the A site and the E site. This P site is involved in the binding of peptidyl tRNA. A site is involved in the binding of amino acid tRNA, and E site is involved deacylated tRNA. Let us have a look that this translation process comprises of activation, initiation, elongation and termination. Activation means we have to make the tRNA charged with suitable amino acids. This is activation. How it occurs? It occurs with the help of amino acid tRNA. It is carried on in a 2-step process. First step is formation of amino acid AMP, and second one is the transfer of this amino acylated AMP onto the tRNA, leading to formation of charged tRNA. Next step, you can see here that this enzyme that carries on the charging of the tRNA is called amino acid tRNA synthetase. Initially, the amino acid binds there, then we have got ATP molecule, and this leads to the addition of AMP molecule along with the amino acid. And finally, when tRNA enters this pocket, there is transfer of this amino acid from this pocket to the tRNA, leading to formation of amino acid tRNA, which is the charged tRNA. For 20 different amino acids, we have got 20 different amino acid tRNA synthetases. Let's have a look that what happens during initiation. A small ribosomal subunit binds to the mRNA, and binding of this mRNA onto this smaller ribosomal subunit leads to incoming the initiator tRNA comes to the P site. After that, another charged tRNA comes onto the A site, and there is peptide bond formation between the amino acids. And finally, the amino acid is transferred from the P site to the A site, and due to the translocation of the ribosome, we will see that these polypeptides are reaching the P site, and at the end point of translation, a complete polypeptide is released. Next, you will see here that what actually happens in initiation. We have got this start codon in the mRNA. This is the smaller ribosomal subunit. The formyl methionine tRNA is entering the initiator site, and onto the P site it is entering, and next you can see here that apart from this, we need certain accessory factors which are recycled. Now the question arises that how the mRNA will recognize, or how the ribosomes will recognize the start site, or the start site is called AUG codon, how it is recognized. For this recognition purpose, you can see there existence of Shine-Dalgarno sequences. These Shine-Dalgarno sequences are present within the mRNA, and it is the hydrogen bond formation between this 16S rRNA component of the smaller ribosomal subunit and the Shine-Dalgarno sequence that helps the ribosome to recognize the first start codon. Next, you can see here that once this start codon is being recognized, we need the involvement of certain initiation factors called initiation factor 1 and 3 and initiation factor 2. What at all they are doing? You can see that the 30S subunit binds to the mRNA taking help of initiation factor 1 and 3. Then initiation factor 2 brings tRNA to the P site, and finally the initiation factors are released so that the larger ribosomal subunit can come and bind to the smaller ribosomal subunit, leading to formation of complete initiation complex. You can see here that initiation in bacteria needs 30S subunits and certain accessory factors, and special initiator tRNA is needed to initiate polypeptide synthesis in case of prokaryotes, that is called formyl methionine tRNA. And this formyl methionine tRNA, you can see here that formylation is done with the amino group present in the methionine residue which is attached to the tRNA. And who is carrying on the formylation? 10-formyl tetrahydrofolate is responsible for the formylation process, and this formylation is carried on in a specialized tRNA that is called as the initiator tRNA. What is so special about it? That it has got 3 GC base pairs at this point, which makes it a specialized initiator tRNA, and it has got this AC bond absent, which is responsible for formylation. These two features in this formyl methionine tRNA or initiator tRNA is responsible for attachment of formylated methionine as well as for the entry of formyl methionine tRNA onto the P site. Rest of the tRNAs enter the ribosome through the A site. You can see here that IF2 binds the initiator formyl methionine tRNA and allows it to enter the P site on the 30S ribosomal subunit. After that, the 50S subunit joins the smaller ribosomal subunit only when the initiation factor 1, 2 and 3 are released. Next, you can see here that if I compare it with the eukaryotic transcription initiation, where the smaller subunit is called 40S, therein we don't have such Shine-Dalgarno sequences in the mRNA. Rather, there we will see that initially we have the involvement of methylated mRNA. The mRNA is methylated and the initiator codon that lies downstream of this methylated cap is considered as the first codon or transcription initiation site. Herein we will see that the small subunit binds to the methylated cap and it scans over the mRNA and reaches the first codon. And you can see here these sequences are called internal ribosome entry site, a eukaryotic messenger RNA sequence that allows a ribosome to initiate polypeptide translation without migrating from the 5' end. Now you can see the details of this initiation process. Herein you can see that initiation factors that are required for all stages of initiation include binding of the initiator tRNA. So initiator tRNA initially binds to the smaller ribosomal subunit. This 40S subunit attachment to the mRNA. You can see here that the cap binding proteins, there are certain factors called 4A, B, E and G which are migrating, and this finally leads to the binding of charged tRNA bound onto the ribosome and attachment of the mRNA. We can see here that the start codon is being recognized taking help of iris sequences. Herein, this complex is called 43S complex, and finally, when this initiation factors are released, the larger ribosomal subunit binds to it. You can see here that we have got eukaryotic initiation factor 2 binds to the initiator methionine tRNA and GTP, and it forms a ternary complex and binds to the 40S subunit before it associates to mRNA. Herein we see a difference. In case of prokaryotes, we have seen that the mRNA is first bound by the ribosomes. Here the story is a bit different. A cap binding complex binds to the 5' end of the mRNA prior to the association of the mRNA with the 40S subunit. You can see here, if I compare these two between prokaryotes and eukaryotes, in prokaryotes there is Stein-Delgado sequences which helps to recognize the first AUG codon, and the mRNA binds to the ribosome, or ribosome binds to the mRNA first, then comes initiator formyl methionine tRNA. It binds to the AUG forming hydrogen bonds. On the other hand, in case of eukaryotes, we can see here that initiator methionine tRNA initially binds the ribosome, not the mRNA. Then comes mRNA and it forms hydrogen bonds with the initiator tRNA. Here another significant difference is that the initiator methionine tRNA is not formylated, but obviously it is different from the other methionine tRNAs. Then we will see the rest of the steps are same, that the larger ribosomal subunit comes in the last part of translation initiation. Now we will be discussing about the translation elongation process. Translation elongation is started by the factor elongation factor 2, which is a G protein and it binds GTP molecule. What it does, that elongation factor 2 GTP complex binds to the amino acyl tRNA, and it helps in the binding of amino acyl tRNA onto the A site, as you can see here. Next, that elongation factor 2 basically recycles between GTP bound state and GDP bound forms. The next step will be the peptide bond formation between the amino acid which is present onto the A site and the polypeptide which is present onto the P site. You can see here that this process is being carried on by an enzyme called peptidyl transferase. Who carries on this peptidyl transferase activity? The 50S ribosomal subunit has got this peptidyl transferase activity. Herein we can see that rRNA is acting as an enzyme. We come to the concept of ribozymes. After that, the peptide bond formation is being done. It means you can see here that the peptide bond formation is taking place and the amino acid is getting transferred from the P site to the A site. This leads to the formation of a site containing the polypeptide chain, and the P site is free, and this P site, which has already transferred its polypeptide chain, means it has got a deacylated tRNA is transferred onto the E site, and as ribosome is moving, we lead to the synthesis of a new A site which is vacant, and the P site is having the polypeptide chain. Now, this translocation process is an essential step in the carrying on the translation process. You can see here that as the ribosomes are moving, we can see that how this tRNA has transferred its polypeptide from P to A site, and after that the ribosome has moved one step ahead and this uncharged tRNA has migrated from P to E site, and this charged polypeptide containing tRNA which was previously residing on A site now reaches the P site, which creates a vacant seat or new place for a new amino acyl tRNA to come and bind onto the A site. This is how the translation proceeds, and you can see that translocation requires another elongation factor called elongation factor G, which also has got a bound GTP molecule on it. So, the two important translation elongation factors are elongation factor 2 and elongation factor G, which are mutually exclusive. Next, you can see here that elongation factors have alternating interactions as GTP hydrolysis takes place, leading to the formation of elongation factor 2 GDP. Next, you can see the entire cyclic pathway of the translation process, which initiates with entry of initial tRNA onto the P site. The next tRNAs come through the A site. After that, there is peptide bond formation, and after peptide bond formation, there is transfer of polypeptide from P site to the A site, which leads to the translocation of the ribosome, wherein the uncharged tRNA is coming out of the ribosome through the exit site or E site, and the polypeptide bearing tRNA gets transferred from A site to the P site, again generating a fresh and vacant A site for further entry of another charged tRNA from the cell. So this is how the translation process goes on. Now this translation is going on until and unless it reaches the stop codons. What happens when it reaches a stop codon? The three stop codons are UAG, UAA and UGA. For these three stop codons, we don't have any amino acyl tRNAs. Rather, release factors are there which come against those stop codons and binds to the A site. And after that, we have got the involvement of ribosome release factors. How they are working? Let's have a look. That we have got release factor 1 in prokaryotes, which recognizes the stop codons UAA and UAG and signals to terminate the polypeptide translation. On the other hand, release factor 2 recognizes UAA and UGA and it signals to terminate polypeptide translation. We have got another release factor called release factor 3. What it does? A polypeptide translation termination factor related to the elongation factor G, and it is responsible for the release of class 1 and class 2 release factors, and it is also responsible for disintegration of the ribosomes into larger and smaller subunits. So you can see here that the release factors are binding onto the ribosome, and after that the ribosome release factor, it enters the A site. It is responsible for release of the polypeptide chain, and it is also responsible for disintegration of the ribosome and disintegration of the entire translation machinery to reinitiate the process again. So herein we have compared the translation machineries of prokaryotes and eukaryotes. The most important difference between prokaryote and eukaryotic translation machinery is that the prokaryotes have got polycistronic mRNA, means from one mRNA we get the translation of more than one proteins, whereas in case of eukaryotes the message is monocistronic, means one mRNA is coding for a single protein. In case of prokaryotes, the second important distinction is the coupled transcription and translation, as because they are occurring in the same place. On the other hand, in case of eukaryotes, transcription is occurring in the nucleus and translation is occurring in the cytoplasm. So there is no question of coupling of these two processes. Next important difference is that we can see here the formation of linear polyribosomes, whereas in case of eukaryotes there is formation of circular polyribosomes. In case of prokaryotes there is no capping in the mRNA, whereas in case of eukaryotes the 5' end of the mRNA is capped. The start codon in prokaryotes is just after the Shine-Dalgarno sequences. In eukaryotes there is no existence of Shine-Dalgarno sequences, and the first amino acid in prokaryotes is a formyl methionine, whereas in case of eukaryotes it is a normal methionine. The ribosomes of the prokaryotes are 70S, which is made up of 30S and 50S subunits, whereas in eukaryotes it is 80S ribosome, which is made up of 40S and 60S subunits. To further distinguish, the small 30S ribosomal subunit is made up of 16 srRNA and 21 proteins, whereas the smaller 40S subunit is made up of 18 srRNA and 33 different proteins. The larger 50S subunit comprises of 23S and 5S rRNA and 31 proteins. On the other hand, the larger 60S subunit in eukaryotes is made up of 28S, 5.8S and 5S rRNA. Along with it, there exists 49 proteins. The elongation factors in prokaryotes are elongation factor 2 and elongation factor G. Herein, the elongation factors are indicated as eukaryotic elongation factor 1, which has got 3 subunits, and eukaryotic elongation factor 2. There exists 3 initiation factors in prokaryotes, namely initiation factor 1, 2 and 3, whereas in eukaryotes there exists multiple initiation factors, namely eukaryotic initiation factor 2. It contains 3 subunits, eukaryotic initiation factor 3, then eukaryotic initiation factor 4 has got 4 subunits, and eukaryotic initiation factor 5. In case of prokaryotes, you can see that it shuts off by dimerization of ribosome in non-growing cells, whereas in eukaryotes the control is done by initiation factor sequestration. So this is how we have discussed and gone through the translation process in prokaryotes and eukaryotes, and we have understood that this process is tremendously conserved in between bacteria and eukaryotes, having minor differences in the initiation process. Thank you.